Hello everyone and welcome to Longevity Article Review number 3. My name is Jordan Marsh and let's get into it. First, let's start with bird nerd time. This is the bar-headed goose. The bar-headed goose is famous for the altitude at which it can fly. It can fly at about 26,000 feet. Comparatively, the Mount Everest base camps are at about 16,000 feet and are famous for people having an inability to breathe or having a really hard time breathing just at 16,000 feet. I'm from Salt Lake City, Utah, which is at 4,000 feet, and then of course sea level is 0. I thought this was an interesting bird to talk about today because a lot of the adaptations that it has in order to survive altitudes like that are actually being used to help in human medicine. So there's this article from the New York Times that says these high-flying geese are the astronauts of the bird world. They can migrate above 26,000 feet, and a better understanding of their efficient use of oxygen could have implications for human medical treatment. I thought that's pretty cool, and we're going to be talking about the way in which we use oxygen in our bodies with this research article today, so I thought this was a cool one to start with. So, bar-headed goose, check it out. So this study basically just investigated the quantity and the quality of mitochondria as it relates to increasing amounts of exercise. So it's very similar to Longevity Article Review number 2, where we had differing levels of exercise and we were measuring different outputs from that. So let's get started. Here are the 4 different groups that they had: an active trained group, a well-trained group, a highly trained group, and an elite trained group. And I'm just going to briefly tell you the VO2 max of each of these groups to give you an idea. So the active trained group had a VO2 of 51 plus or minus 4. The well-trained group had a VO2 of 64 plus or minus 5. The highly trained group had a VO2 max of 71 plus or minus 2. And the elite trained group had a VO2 max of 77 plus or minus 3. All of these are pretty high, so just remember that. These are not necessarily sedentary individuals. Even the active trained group, being the lowest VO2, is a fairly active person. Each of these groups had 6 people, and basically what we did is we just brought them in, we tested their VO2 max, we put them into different groups, and then we did a muscle biopsy. So they took a muscle biopsy from the vastus lateralis muscle that's in your quadricep, and basically just did a bunch of tests on their mitochondria to see the quantity and the quality of the mitochondria. So here are the subject characteristics. They did a pretty good job of keeping the age well contained. And again, this is another study that has mostly males. There's 23 males in this study and 1 female. So keep that in mind as a potential limitation of the study. So here's the results. This is a graph of citrate synthase activity. Citrate synthase is an enzyme on the surface of mitochondria that is used as a biomarker for mitochondrial density or mitochondrial content. So here's that's what it says right here. Basically, this just means, the higher trained you are, the more well-trained you are, the higher your VO2 max is, the greater density of mitochondria you have in your cells. And specifically, as you can see here, this is a significance marker, and it's measuring the significance based off of its comparison to the elite trained athletes. So we know that there's a statistically significant difference in the mitochondrial content in elite trained athletes compared to well-trained athletes and compared to active trained athletes. So that's an awesome thing to know. We have more mitochondria the more well-trained we are. But the next part, I think, is what's really interesting. So in order to understand this, I'm going to show you a graph. There's going to be 2 graphs. There's going to be a graph A and a graph B. But before I showed you the graph, I wanted to give you an idea of what we're looking at. So here in columns are each of the subjects in their different groups. And in rows is the graph A and then graph B. So row A is measuring the difference in mitochondrial activity, and it's not necessarily normalized to the amount of mitochondria in the tissue. So that's what I try to show here, is that in the active trained group, maybe they have less mitochondria than the well-trained group, maybe they have less mitochondria than the highly trained, and then the elite trained group. So it's really not normalizing based off of, how many mitochondria are in the cell. And so That's what B tried to do. So as you can see here, what B is saying is we actually want to measure the quality of the mitochondria rather than the quantity of mitochondria, okay? So I'm going to show you the graph. It's very busy, so don't get too freaked out. And I'm going to try and go through this little by little. So as you can see here on the x-axis, there's these different letters. All that means is it's just different mitochondrial energy states. And each state reflects a different aspect of mitochondrial function. So there's just different states, and they're measuring the ability of oxygen to be utilized in that state at different times. And we are comparing it for each of the 4 groups, okay? Remember, the graph A here is measuring it just straight up. This just reflects the overall mitochondrial capacity of the tissue, and we are not normalizing it for how many mitochondria are in the tissue. So as you might expect, because we know that elite trained athletes have more mitochondria in their tissue than those that, like an active trained athlete, we can see that there are many different there's many different significance markers between the groups. However, in B, we divided all of that by the citrate synthase content, which basically just meant we want to compare mitochondria to mitochondria. We only want to see how well a singular mitochondria compares to another singular mitochondria in another athlete. And so you can still see here, though, that there are some significant markers here that show that, yes, not only do we have a higher quantity of mitochondria in our tissue, we also have a higher quality of mitochondria in our tissue, the more we are well trained. So I think that's a really cool finding and something that this study does a good job of showing. So here's 3 good things that we can take away from this. Of course, there's enhanced mitochondrial quantity the more we engage in aerobic fitness, and that's helpful for our ability to utilize oxygen and ultimately helpful for our ability to have healthier cells that are able to generate more energy. We also have improved mitochondrial quality. Higher levels of aerobic fitness, actually just mitochondria for mitochondria, can say that we are better able to utilize oxygen. And then we have these comprehensive adaptations, which says that we can actually improve our quality and quantity the more and more we exercise. So really good things to be aware of. Here's 3 things to consider, maybe some limitations of this study. So the sample diversity, I mentioned it again, there was 23 participants that were male and just 1 that was female. So that may limit how well we can generalize these findings to the overall population and specifically to females. So this study is very cross-sectional, and that we just did a biopsy at one point in time and tried to see differences in mitochondrial content and quality. And so it may be difficult to establish this causality between aerobic fitness and mitochondrial adaptations, but certainly there's a correlation there. So the training variability is certainly something to consider here as well. Again, we didn't put each of these subjects through the same exact exercise workout routine. We just said, come on in, we'll test your VO2 max, and then we'll split you up into groups. And so maybe there's differences in the training regimens that these different athletes did that might not only be attributable to their VO2 max, might be attributable to their weight training or to their stability training or something like that. So maybe something that we should keep in mind when we consider this study. All right, thank you so much. I will see you at LAR number 4.